Okay, so what this next video is going to go through, it's going to go through what we call UV visible spectroscopy. So it's kind of a subset or another thing that's very, very similar to colorimetry. So colorimetry is based on color, yes, and the color increases. UV visible spectroscopy is actually not just based on, so colorimetry is just about visible light, whereas UV visible spectroscopy actually takes us down a bit further. Now, if we think all the way back here to the electromagnetic spectrum, so colorimetry only corresponds to this bit down here, whereas UV vis actually also includes wavelengths in the ultraviolet spectrum as well as the visible spectrum. So it's kind of like a bit broader in terms of what we can find out from this. So having a look here, we've got a bunch of components that make up the UV vis, we call it UV vis spectroscope. So what you do is you have this light source. Now this light source emits both UV wavelengths and also visible light wavelengths. Now what happens is you kind of focus it through this prism monochromator and they bring together the light and focus it really, really strong, just down to almost a single beam. Now, so you've also got a slit here. Now this slit means we're only going to capture the light rays that are getting through here. These ones here are going to get kind of absorbed by the black plastic. So then we got to focus it even more using the monochromator. And then after we've focused it with all these lenses, is it's going to pass through this little, and usually they're plastic, these little things called a cuvette. Now a cuvette is the substance. So in here is your substance to be tested. So inside the cuvette, you would put your substance to be tested. So what that means is you pass the light through that substance and whatever doesn't get absorbed, so this is transmitted wavelengths, yeah, whatever doesn't get absorbed will get detected by this light detector, and then we just put it as an output, usually digitally, not so much old school kind of needle anymore. So that's kind of the basics of the spectroscopy machine. And first off, this video is going to look at how we can use the spectroscopy machine to give us qualitative analysis. So what we find is that a particularly coloured substance, say a green leaf, yes, will absorb a range of wavelengths in a certain region of the spectrum, and it will reflect or transmit certain wavelengths as well. So particularly in this, we're looking at transmission because we're looking at what passes through the cuvette so that it can be captured by the detector here. So what we can do is we can actually figure out the wavelengths that are absorbed and how much is absorbed at those particular wavelengths. So that's done by how much light makes it through when you shine particular wavelengths through your sample. So what we do is we graph the absorbance versus wavelength. So you would see that in chlorophyll A is the pigment that we're analyzing here. And you can see that it absorbs almost 100% of the indigo wavelength. Sorry, I was doing a backwards rainbow for a minute. It also absorbs about 75% of the orange and red. So we're taking out some of the purple, we're taking out some of the orange and red out of this light. What does that mean? These wavelengths, in particular, you can see this green here, is passing straight through. So what I would say is that this compound, chlorophyll A, is going to appear green. Yes, it's going to appear green because, so it's going to appear green because the green wavelengths are the ones, because green wavelengths have passed through, have been transmitted. Yeah, nice and easy. So you can see here is spinach chlorophyll, very similar again. So what you can do is you can actually look at these particular graphs, and each graph will be unique for a particular substance. So once it's unique for a particular substance, then we can use it for a particular substance. We can use this for identification. So if it follows a particular pattern, we go, that's chlorophyll A. Here's an unknown pigment, let's run it through. It follows the pattern of a carotenoid. So we know that it's a carotenoid. And so that is how we can use, here's just another example of how they might do it. You can see here, so what we can do is we can actually use, this is quantitative analysis, sorry, wrong one there. So what we can do is we can use this to identify what unknown pigments are in a particular solution.